I'm going to talk about Macular Telangiectasia Type 2, MacTel, retinal disease. Progression of this retinal disease will affect central vision of MacTel patients. They will start losing their central vision. Medium rare disease, what we call now an uncommon disease, prevalence to be estimated between 0.1% and 0.045%. Median age of diagnosis in late 50s, beginning of the 60s. No difference that we have been found between males and females so far. But an interesting comorbidity that we have seen in MacTel patients is that a lot of MacTel patients also present with type 2 diabetes. They are also often smokers, they have high BMI, and they are at risk of hypertension. A very interesting feature of MacTel is that it's been observed to be a familial disease. So not completely penetrant, but we have seen that patients affected with MacTel disease oftentimes also had relatives also presenting with this disease. In their eyes, they present lots of different phenotypes that go wrong in their eyes. I'm not an ophthalmologist, so I'm not going to try to explain what's going on, but this is just like a fun graph to show you that there are lots of different things that's happening in the retina, mostly with leakage on the vessels and the capillaries that start happening. There is degenerations of the layers within the retina. We have crystals, deposits, we have breaks in the layers, lots of different things going on. And it's quite of a slow progressing disease. So yeah, what I'm going to show you now are a series of studies that we have performed that basically got us from not knowing much about this disease and then knowing maybe potentially a mechanism that goes from the basic genetics all the way up to the disease. And I'm going to show you what we did. So the first was a GWAS study that we published in 2017. This was a genome-wide association study in which we collected more than 700 cases and almost 2,000 controls, measured SNPs on an omnichip, and then we imputed them, and we ended up with 6 million SNPs. And then we performed the very first GWAS. Now, in this GWAS, we found 3 genome-wide significant loci, which I'm highlighting in here, and other 2 loci which were suggestive significant, and they replicated on an independent cohort. We also estimated heritability on this study, and assuming a prevalence around 0.1%, heritability of MacTel was 0.74, which is quite high for a disease that is of this rarity. An interesting thing that we found in the first GWAS is that 4 out of the 5 signals that were found and replicated were actually all connected to the glycine and serine pathway, metabolic pathway, when instead another one, the one that I'm highlighting here in pink, was this intergenic SNP close to the gene TMEM161B, and it was sitting on this pseudogene MIR9-2. And this SNP has been previously identified to affect retinal caliber, so the vascular caliber of vessels in the retina, and also retinal thickness. So we had a GWAS with almost like 2 signals, one which was a metabolic signal, and another one which was a more localized signal that was affecting the retina. Yeah, so 4 of these were part of the glycine and serine pathways because these genes were found to all connect to these 2 amino acids. So the first question we asked was, can we find any differences in these metabolites in blood of MacTel patients? So we performed a quick metabolomic study in which we took 50 MacTel cases and 50 controls. We measured 850 metabolites in their blood, and we performed a very quick metabolomic study. And guess what? The first 3 metabolites that we found, the 3 most significant metabolites that we found, were indeed glycine, serine, and threonine. This was another metabolite next to them. And what we found is that all 3 of them were depleted in MacTel cases compared to controls. And we weren't particularly surprised to find all 3 of them because they're all very connected in what is called the glycine, serine, and threonine metabolic pathway. So they're very, very close to each other. So with the first study, this first GWAS study, what we found is that we had genetic loci that were associated with the disease. They were pointing to a potential metabolic hint that was affecting this disease. And we also found metabolites, these same metabolites were associated into a study to this disease. So the first question that popped in mind back then was, great, so are these metabolites maybe causing this disease? So is a depletion of this metabolite causative for MacTel disease. Now, a way to do this is to perform Mendelian randomization. So we perform the Mendelian randomization study. And for those of you who might not be familiar, I'm not going to go into detail about it, but basically Mendelian randomization is based on using SNPs as instrumental variables. And very, very simple idea. Oh, this looks awful, sorry. Is about using multiple SNPs to basically predict an exposure and then see whether all of these SNPs can also predict the outcome. And if that's the case, you start to assume that maybe there may be a causative mechanism between this exposure and your outcome. First thing, however, we had to do was to find the instrumental variables that needed to be used to perform Mendelian randomization. So what are the SNPs that can predict these metabolites, right? So we're interested in glycine and serine. What are the SNPs that can predict these metabolites? So we entered in collaboration with Cambridge University, and we published this big study. Well, they published this big study. We're just collaborators out of it, that we're looking at basically MQTLs. So metabolite QTLs, and this is a snapshot of the results. In here, you have basically the genome-wide and metabolome-wide, and each of these towers is one of those associations between a particular locus and a metabolite. So using this study, basically, we could find all of the different SNPs that affect different metabolites, and we could use these SNPs as instrumental variables. So once you do that, you can basically, you can collapse SNPs into either polygenic scores or connect the summary statistics of these genes, of these MQTLs with your GWAS, and try to almost genetically predict the level of these metabolites into your patients. So if you have genetic data on the patients, you can predict that. And when we did this, we saw that serine and glycine were indeed by far the two metabolites that were the mostly associated via Mendelian randomization with MacTel, potentially strengthening the hypothesis that these two metabolites might actually be causative for MacTel disease. Very interesting result we found out of this analysis is threonine. Look at where threonine ended up being. Threonine was very non-significant into this. That was also like a very nice way of saying, okay, maybe threonine was only associated with this disease in metabolic data because these metabolites are so close to each other, but their genetic basis is different, and the genetic was not confirming a causative role of threonine on this disease, which was quite interesting. Now, we found also many other metabolites that survived multiple testing, and they were still significant, apart from glycine and serine. One thing that I want you to focus on, very interesting, is look at the odds ratio and the effect size of Mendelian randomization performed on glycine and serine. They were also negatively associated. So even just looking at the genetics, we could tell that these patients were meant to have lower level of glycine and serine, which is exactly what we found in their blood by their metabolomic study. Then we have many other metabolites that were associated as well. However, these metabolites somewhat share some of their genetic signal with glycine and serine. So when we conditioned out glycine and serine out of this analysis, nothing remains significant of anything other, apart from one, which was alanine. This is another amino acid. There's not much to do with glycine and serine. And interestingly, this one was positively associated with the disease. So far, we have no idea what alanine was doing to this disease. We just took it as face value and were like, sure, something else might be causative associated with this disease, but we have no idea why that would be. One thing that I want to show you, however, is we use this polygenic score to conduct a conditional GWAS. We performed again the GWAS, but we included in the covariates of our model the genetically predicted serine and genetically predicted glycine, and two loci that were previously only suggestive significant now rose to full significance. Now, I'm not highlighting the central peak that you see in there on chromosome 5. That's because that was the chromosome 5 peak, which we already knew was already fully significant and had nothing to do with glycine and serine. So, of course, that remains significant. But other two rose up to significance. One of them was an intergenic gene between SLC4A7 and EOMS. Another one in this sphingolipid gene called SERS4. In that same study, what we also tried to do was, could we potentially use this genetic data and look at phenotypes? So if we only start looking at cases, right, we completely forget the controls. Let's just look at cases. We had this very big data set of MacTel cases, more than 400 cases. We had 5 years of phenotypic data. For phenotypic data, here I mean data that was collected on the eye images. So clinicians take tons of eye images of these patients. And on these eye images, we can actually measure numerical phenotypes, right? That measure lots of different things that happen in their eyes. And we had more than 120 phenotypes, and there is spatial on top of it. So what we thought was the first thing to do, maybe, can we reduce the dimensionality of the study? Can we collapse these phenotypes into a small set? So we created via factorial analysis what we call endophenotypes. So combinations of these phenotypes that we can observe in the retina. And could we use these endophenotypes to understand what's going on? And an interesting thing that we found was that genetically predicted serine, again, the formatting here is very interesting. Genetically predicted serine was associated with lots of these phenotypes in cases only, and in particularly negatively, which meant lower serine, and this was genetically predicted serine, was associated with a worsening of this phenotype in the eye. So people who were predicted to have lower serine were worse off in our dataset. Again, strengthening a potential causative association of this metabolite with the disease and even its own progression, which was quite exciting. We also perform a FEWAS using these endophenotypes, in which we try to cluster the SNPs that were previously found to be associated with this disease. I'm not going to go into the details, but basically what we found is that the serine and glycine associated SNPs were found to affect similar phenotypes, when instead the one in chromosome 5 affecting retinal vasculature was found to be associated to stuff that was completely different. So if the first GWAS, we were left with this mechanistic effect with SNPs associated to metabolite that maybe are associated with MacTel. Now with the latest study, we found that some of these SNPs mostly contributed to indeed levels of only glycine and serine and not threonine. Serine was probably causative associated with MacTel. Glycine was causatively associated with MacTel, but I didn't have time to go inside that. But at that point, we already believed that this causative association was only driven by this very shared deep connection with serine. And also alanine might have been a very important player on this disease, but we had no idea why back then. And that's when, after all of these different omic study, we had a very interesting clinical finding. And with this clinical finding, we were actually able to pinpoint a much more precise mechanism behind this disease. And it all started with a convergence between an hypothesis and a clinical finding that happened basically at the same time. So there was this mechanistic hypothesis that if we take serine, let's see what serine does. One of the things that serine does, apparently, I'm not a molecular or metabolic person, so I don't know exactly all of the different pathways the serine is involved. I'm a bioinformatician. But one of the things that serine is used for is used by this enzyme called SPT. Serine gets ligated with palmitoyl-CoA to create these sphingolipids called sphinganines. Now, if one hypothesis is if serine is extremely low or is not available, what SPT is going to do is going to take alanine instead of serine. And if SPT takes alanine and ligates it to palmitoyl-CoA, it creates this other sphinganine, which is called deoxysphinganine. Deoxy because it lacks the deoxy group. And deoxysphingolipids have been seen prior to actually be toxic for neurons. They have a toxic effect on neuronal cells. Very interestingly, there is this mutation on this gene called SPTLC1, and this is a very, very rare mutation. However, people that are affected by this mutation, their SPT enzyme gains an affinity to take alanine instead of serine. So even if serine is available, it would still prefer to take alanine instead. So people affected by SPTLC1, they have a huge production of deoxysphingolipids because they have this mutated enzyme, right? And people affected by this disease are known to have this neuropathy called HSN1. This is a peripheral neuropathy in which people start losing their feelings in their fingers and in their feet. So peripheral neuropathy. Very interestingly, at the same time that all of these hypotheses started happening, a collaborator of ours from Utah found an extremely young MacTel patient. It was in his 20s. Now, you remember at the beginning I told you that MacTel patients usually are more towards their late 50s, their 60s and stuff. This was a very, very highly progressed patient that was in his 20s. Well, this patient had HSN1. And the same patient said, well, also my father has my same issue, and is actually also having problem with his vision. Well, behold, when we look at their eyes, they had MacTel. So one thing that we tried to do in this study was, can we recruit as many HSN1 patients as possible? And this disease is super rare. So there is very low chance of, if these diseases are not connected, that we're going to find MacTel within HSN1. And when we did look, and you can see in this one, we found that in these three families, people who had HSN1, which is here highlighted in gray, also presented with MacTel disease, which is here highlighted in black, which was an astounding finding, exactly because of how rare both diseases are, finding them to be comorbid, and in such of a high degree was an extremely high finding. You remember at the beginning, I told you we found alanine to be causally associated with this disease, and we had no idea why. And it was because of high alanine that might have been causative associated. Well, this might have been an explanation. So if we imagine maybe a patient with lower level of serine or even normal level of serine, but they have an extremely high abundance of alanine in their metabolism, maybe SPT is going to take alanine and ligate it with palmityl-CoA just because of the ratio between alanine and serine is completely imbalanced. So that fits very well with what we saw so far in all of our studies. We also check metabolic levels. So in MacTel patients, sphinganines, we saw that normal level of sphinganine did not change. However, when we test whether this hypothesis was true in a larger MacTel cohort, they did indeed have higher level of deoxy, again, confirming that deoxy might have been the thing that was driving this. Of course, they had lower level of serine, and this deoxy level negatively correlated with their serine level and positively correlated with their alanine levels, which is exactly what we were expecting out of these results. When we performed my study, here we put mice on a serine and glycine depleted diet. So these mice were in their diet, they had no glycine or serine, and they could not make it from other things. And we saw that their deoxysphingolipids after this diet were increased in their periphery, just like we saw in MacTel patients, but also they had higher deoxy in their retina, in their RPE, which is a layer basically that is behind the retina, is on the back of our eye, and in their sciatic nerve, which is in the nervous system. Additionally, we saw that these mice, by the B wave and the photopic flicker, actually starting to have a phenotype in their eyes in which they were starting to lose some form of vision, and they were starting to respond much slower than other mice that were not in the diet to hot plate. So they were starting to lose their senses on their periphery. Via organoid study, we also saw that exposing retinal organoids to higher level of deoxysphingolipids was toxic. We started observing cell death, and this cell death was, however, we could recover this cell death if we added fenofibrate on top of the deoxy as a rescue, and it was actually rescuing it. So if with our Mendelian randomization, we were left with a mechanistic, maybe clues that looked more like this, with this latest study, it was really when we pulled everything together and we had a pretty good idea of one of the potential mechanisms of MacTel. So we have SNPs, which potentially cause lower serine. This lower serine results in high deoxy, which is toxic for the retina and probably ends up causing the phenotypes that we observed in MacTel patients. However, there are also other mechanisms that may cause this high deoxy, such as very rare mutation, for example, in SPTLC1s, but these are extremely rare, or also an extremely high imbalance of alanine, which also will result in high deoxysphingolipids, also causing this disease. So it was a very clean pathway. However, one of the things that we have observed is that MacTel patients overlapped with controls for the level of deoxysphingolipids and also serine, right? So these metabolites are not enough to completely distinguish between MacTel cases and controls. It's not that every MacTel patient has extremely high deoxy and all controls has very low deoxy. So they're not enough. So what else? What might be lying underneath that we could look at? So we went back to our metabolomic study and we expanded it and looked much further and did a proper one. So here we had 60 cases and almost 60 control, more than 700 metabolites. And we tested for all the metabolites that were different, and we found many metabolites that were different in MacTel cases and controls. We have lots of amino acids, fatty acids, glycerophospholipids, glycerolipids, sphingomyelins, lots of different stuff. So I'm not going to enter much in the results of each single one of them. The first two that, of course, they were the most significant one were the glycine and serine, very much expected, really good. The third most significant metabolite was an interesting one. This is called a 2-hydroxyglutarate. This is a fatty acid. It has not much to do, for as much as we know, with glycine and serine. It's not part of that pathway. However, a very interesting thing of this metabolite is, remember at the beginning, the SNP that we found affected these three genes, the one that we thought were related to glycine and serine, and then confirmed with the MQTL study that were actually associated with the abundance of glycine and serine. This was CPS1, PSPH, and PAGDH. Now, if you look at KEGG, PAGDH is associated with the biosynthesis of 2-hydroxyglutarate. And in my studies, when they knock out PAGDH, levels of hydroxyglutarate plummet as well. Again, confirming this was also the level of 2-hydroxyglutarate was depleted in MacTel patients compared to control. And this for us was a very interesting result. More than from a mechanistic point of view, it was very interesting to see how our metabolomic study was back confirming our genetic findings via almost a separate route, which was an interesting result to find. When we performed the enrichment analysis, and we also tried to look at are families of metabolites completely changed or homogeneously changed between case and controls, of course, we found a depletion of the serine and threonine metabolic pathway. We knew that. However, we found that phosphatidylethanolamine metabolites and lysophosphatidylethanolamine metabolites were upregulated, so all of them were overabundant, and sphingomyelins were quite low. And the sphingomyelins in particular, we found to have a very interesting behavior. When we looked at differential coabundance, this is where we test whether correlation between metabolites are similar in cases compared to controls. What we found is that everything, as you can see from this core plot, looks like very, very similar. However, when you look at sphingomyelin, you start seeing like an interesting patterns. This was the one that was changed the most, and was changed in particular on its relationship between the glycine, serine, and threonine, which was the main pathway that we were interested to, and the phosphatidylethanolamine pathway, which was another one, which was one of the highest, most significantly associated pathway in this study. And if you look at the controls, sphingomyelin was very highly associated with all of them. It had a positive association with the glycine and serine, and a negative association with the phosphatidylethanolamines. When you look at controls, this association was almost gone. It was completely disrupted. So there was another indication that sphingomyelins were low, yes, but they may not have a direct relationship in terms of their association with the disease in terms of abundance, but maybe instead is their role of co-modulating other pathways that are instead associated with the disease. So if with the previous study we were left like this, with this one we added even more stuff. We involved the phosphatidylethanolamines, the sphingomyelins, which again add other sphingolipids, just like the deoxysphingolipids, and other metabolites that might have been associated with this disease. All right, so with these 4 studies, we basically created an entire mechanism. We're like, all right, we know what the main mechanism can be. We know that there are other players. We know that we can go from the basic genetics all the way up to the disease. What if we start digging deeper? What if we expand our study and we start looking of stuff that maybe like we have missed and go a little bit deeper and increase even more our sample sizes? What are we going to find? So we perform what we call our GWAS 2.0 study on MacTel disease, in which basically we doubled our sample size and tried to see what was happening. And we start checking even further. And this was the results. This is the Manhattan plot. And we were very glad to see that there was clearly a confirmation of our initial results, which all of our SNP that were identified in our first GWAS were confirmed, apart from one, which was in chromosome 3, which it was suggestive significant and remained suggestive significant. Instead, the one that you see in chromosome 7, which was suggestive significant, actually become fully significant in this study. We're also glad to see that the two loci that we found via conditional GWAS now rose to full significance, again strengthening their association with this disease. And also we found other genes. Some of them were genes that were almost lying on the periphery of the glycine and serine association. For example, these were transporters, so they were not exactly related to the biosynthesis of these metabolites, but how these metabolites are used and how they are transported. Another one, for example, was SERS4. This was a sphingolipid gene, which we already identified in the conditional GWAS, again, relating to the story of our sphingolipids. And then this locus in chromosome 9, this TTC39B, which we had no idea, we still don't know exactly how this gene is acting on this disease. And it's quite interesting because when you perform EQTL analysis or even a TWAS analysis, we found out that this gene was mostly involved in liver. And again, it makes some sort of sense because now we know that MacTel patients have a systemic metabolic defect, and so the liver is likely going to be impacted by these differences. We also found another gene which was positively associated with MacTel, which was this gene called MXtra7 in chromosome 17. This was by the EQTL and the TWAS, quite strongly associated with MacTel. And this gene had been already seen to affect macular thickness and other macular phenotypes. So it was a gene that fit very, very well with our hypothesis. Again, starting to highlight the fact that in MacTel, we probably have almost two parallel mechanisms going on at the same time. We have a metabolic dysregulation, which is systemic, and then we have a localized, potentially susceptibility mechanism that makes these patients more prone for the retina to be affected by the systemic mechanism. We're also working right now on performing what we call the Metabolon 2.0 study, in which we have more than doubled our sample size. Before it was 100 patients, now we almost have 600 samples. On this, we have many more metabolites. We have 1,100 metabolites. All of our samples come from controls plus more than 300 MacTel patients, 168 type 2 diabetics patients, and 21 HSN1 patients. Remember at the beginning, I told you that MacTel patients have a high prevalence of type 2 diabetes. So this was our chance to start digging also deeper about the relationship of diabetes and MacTel, and also look at a much more broad range of metabolites in HSN1 patients, which we know are related to MacTel because of that one class of metabolites, but what's going on with the other ones? So, and how do they relate to each other? And when we performed, of course, like linear regression to test all of these metabolites, correcting for different covariates. And we test all three diseases for these metabolites, and we corrected for, they were all included in the same model. So the presence of one disease, the effect of one disease was corrected by the presence of the other two diseases in case, because we know there is high overlap between the three of them. And it was very interesting to see that the number of significant metabolites that we found was almost reflective of the complexity that we know happens in these three diseases. So HSN1, which is a very, very rare disease, very Mendelian disease, 2 or 3 loci, one of them is completely causal, fully enough to give you your periphery problem. Only had around 60 metabolites that were significant. But also it had a lower sample size so far. MacTel, which is an uncommon disease with a little bit more loci that we have found to be involved. So medium complexity disease had around maybe like 150 metabolites that were involved. And T2D, which actually has less samples than MacTel in our study, so technically should be underpowered, instead had almost 500 metabolites that were different. And T2D, we know it to be quite common, extremely complex disease. Latest GWAS on type 2 diabetes have identified more than 200 loci. So it was very interesting to notice how this metabolic study or how the metabolic study can almost recapitulate metabolically the complexity of these diseases and see like how simple and clean diseases like HSN1 have much lower signal than much more common, much more complex diseases like type 2 diabetes, in which lots of things are completely changed. A very interesting things that we noticed when we compared the significant results about all these three diseases was that, first of all, only 4 metabolites were significant and in the same direction for all 3 diseases. These 4 metabolites were 2 deoxysphingolipids and 2 sphingomyelins. Now, MacTel patients, HN1 patients have a neuropathy because that's how they are defined. MacTel patients, I don't have time to present it today, but we are starting to see a generalized peripheral neuropathy also in MacTel patients. And these are even MacTel patients who don't have HN1. They have a peripheral neuropathy. They're starting to lose their sensitivity in their fingers. Or they report symptoms that are highly associated with peripheral neuropathy. And type 2 diabetes is very well known to have a problem related to peripheral neuropathy. And guess what? The only phenotype all these 3 diseases share is peripheral neuropathy, and the only metabolites they share are the one known to cause peripheral neuropathy. And we could see that from a metabolic study. Very interesting thing to notice is that our HN1 and MacTel almost did not overlap. However, almost half the signal of MacTel overlapped with type 2 diabetes. Remember how I told you that type 2 diabetes and MacTel are very comorbid? Lots of MacTel patients present type 2 diabetes, but not many type 2 diabetics present MacTel. In fact, again, you can see from the metabolomic study, only a very small fraction of type 2 diabetes is included into the MacTel signal. So there is a high prevalence. Also, we perform genetic correlation between the 2 diseases. This is where basically we try to correlate whether the GWAS signals go in the same direction. We found that there was a positive association with it. When performing Mendelian randomization on the effect that type 2 diabetes might have on MacTel disease, we found another significant association between the 2, and it was positive. And very interestingly, there is a retinal disease that often happens in patients suffering from type 2 diabetes, which is called diabetic retinopathy. Very interestingly, we found almost no diabetic retinopathy in MacTel patients that have diabetes. So it looks like that there is no overlap between this retinal disease that is very common in diabetes with MacTel patients with diabetes, which is quite intriguing. When we look at the effect that, so here I'm trying to understand whether, okay, we know that half of the MacTel signal of metabolites overlaps with type 2 diabetes in terms of significance and direction. However, when we look globally at the entire signal across all of the metabolites that we have tested, do they have the same signal? Does the signal metabolically correlate across the 2 diseases? Yes. When we look at each of these points here is a metabolite. Forgive me for like the labels of the metabolites themselves are too small, but that's not the point. When we plot the effect that each of these metabolites had on MacTel on the x-axis and the effect that the same metabolites had on T2D, they are similar. They go in the same direction. They are extremely correlated. Remember, this is not because our study is biased because MacTel patients also have diabetes, not because they are corrected. Those 2 terms are included in the same regression, so they take care of each other. But still, they remain correlated, which is fascinating. And especially it's fascinating compared when we look at the the relationship between MacTel and HSN1. We saw these 2 diseases that globally are not metabolically correlated at all. The only thing that they share is what I'm highlighting on top there that is positive for both of them, which are the deoxysphingolipids, which is the only thing that we know they should share. And in fact, is the only thing they share. HN1 looks nothing like MacTel metabolically, apart from that very particular set of metabolites and some sphingomyelins, which are negative in both. The same study, the next thing we tried to do was to try to divide patients given their metabolic profile. We performed clustering analysis on the metabolites of all of our patients in that study, all of the 600 patients, whether they were MacTel, type 2 diabetes, control, HSN1. we performed this clustering analysis using the metabolites that were significant for MacTel disease. And when we did that, we found there were, I here divided it in 6 groups. Most of you know that clustering analysis is more of an art form than a strong and robust science. So again, very debatable whether here there are 6 groups, 3 groups, or even just 2. But the very interesting thing to notice is that there is clearly a group of that has a lot of MacTel patients and is clearly a diabetic group. This is all on the left. If you look at the group on the right, the 2 groups on the right, they have a lot of MacTel patients. However, they have no type 2 diabetes. And then there is a group in the middle who still has a lot of MacTel patients. Of course, metabolites are not the best predictive things, but much less than the other 2 groups. And however, is the most of the HN1. So this group contains the controls and the HN1. So even the metabolites were already ready to divide our MacTel patients into 2 groups, one with diabetes and one without. And when you look at, so we also looked at rare variants, didn't have time to go down and present this, but we also performed all exome sequencing and all genome sequencing on most of these patients. And when we grouped rare variants that were part either of the glycine cleavage pathway or the serine biosynthesis pathway, we found that the high type 2 diabetes group was actually enriched with variants in the glycine cleavage pathway, when instead the MacTel group without diabetes was enriched in the serine biosynthesis pathway. So metabolic groups also divided rare variants that we could find in the WS and WGS. A question that we always get, which is quite natural, is if serine and glycine and the deoxysphingolipids are so related with this disease, could we supplement MacTel patients with glycine and serine and look at what happens? And this is what we're trying to do here. This is a very preliminary study in which we're still in recruitment. Actually, no, the recruitment has just finished, to be precise, but we have recruited MacTel patients, and to these MacTel patients, we are giving them supplementation of serine. We have different arms: high serine, low serine, fenofibrate alone, or serine plus fenofibrate, which we know reduces deoxysphingolipids. We're going to follow them for 10 weeks. 10 weeks is not going to be a long enough time to evaluate any efficacy on the disease itself. The disease is very slow progressing, and we're not going to see any changes in their eyes. It's way too quick of a study. But what we could check is, can we use this as therapy? And is this therapy potentially going to be effective on the level of deoxysphingolipids? So can we actually raise their level of serine, and can we reduce their level of deoxysphingolipids even in such a short amount of time, and how safe it is? And to do all of this, of course, we are collecting metabolome data, so all of their metabolites. We're performing blood labs, and we're also collecting their microbiome, so we will perform some microbiome studies. Other things we're doing in the periphery, we have WGS, in which we're looking at the rare variants. We're doing a mitochondrial GWAS to check whether there's a mitochondrial signal. Seems like there is, very exciting. And we're doing yet another GWAS, which we will call the GWAS 3.0, in which we are doubling once again the sample size. All right, so to summarize what we found so far for MacTel, MacTel is an heterogeneous and uncommon disease. Heterogeneous because we see a plethora of different phenotypes and clinical signs in the eyes. MacTel is a medium complex disease with a few Mendelian variants. Now, I think a lot of us that work with complex diseases, especially in genetics, are quite familiar with this plot, in which we usually divide the relationship between variants that are extremely rare and they have huge effects on diseases, and usually diseases that are completely Mendelian in nature tend to have those variants that are up there on the top, or very common disease and very complex diseases, which are riddled with very common variants with very, very small effect, and their mechanism is quite complex. MacTel truly lies in between. What we believe is that we have a disease that is this medium complexity disease. It is a few mechanisms which are not exactly the same, but they're similar, but it's not too complex, but it's not too rare. And it also fits with how common it is this like uncommon disease. So it's not extremely rare, but it's not even a common disease, like, for example, another retinal disease, which is age-related macular degeneration. We believe we have a systemic metabolic defect, which may be or is probably coupled with a localized retinal susceptibility. We have more than just retinal manifestation. So MacTel patients have problem in their eyes. However, we started noticing that they have problem with their sensory phenotypes, as well as systemic metabolic defect. We have potential molecular subtypes of MacTel. Are the MacTel patients with developed diabetes similar to the one that don't? Or are the MacTel patients with HN1 similar to the one that don't have? So far, we see that HN1 is very metabolically different from the rest of MacTel. So they might actually be almost different sub-diseases. And this disease seems to be the perfect candidate to keep going with omic studies. We're also, just this FYI, at the Lowy Medical Research Institute, we're also going out now of our comfort zone. We're stepping out of MacTel. We're looking at other diseases that are more general, such as AMD, diabetic retinopathy, and glaucoma. So all of these studies are all upcoming, and we're looking at all of those. And with this one, thank you everyone for coming and for attending. I'm happy to take some questions. And we are also hiring. So if anybody is looking to work on retinal diseases and omic studies, let me know. Contact me, contact us. It will be fun to have you come on board. Cool. Thank you very much.
Do we have any questions? So let's take your word. I'm curious about a few tangents. One of them is threonine, actually, even though you dismissed it initially as like not having correlation, but in the metabolism, the glycine can be generated from serine and threonine. So they appear from different pathways.
Yes.
And glycine is actually essential for anything related to metal transfer, and is implicated in different disorders related to both lipids and but most importantly histone methylation and all the epigenetics. So that's the connection right there. For that reason, I was curious if maybe with new data you can like see, because they're well known threonine depletion diseases which have similar manifestation, because it also converts to glycine contributes methyl to all these other areas. So that's one question. Another question is like, when your supplementation, you assume that across the blood-brain barrier goes where it's supposed to go, or
the only assumption we have on that is that with mice we can see that a systemic depletion of glycine and serine results in a change in the actual retinal tissue. So we don't know whether the changes that we're going to make with the diet in humans are going to have an effect on the retina, nor unfortunately we can measure it directly in humans yet. But given what we saw in mice, we saw that a diet change manages to affect the retina in mice, but it's a good question. We don't know the general obesity between the two species. Yes.
Usually there's a short list of acids which are transported, and serine is not in it, but there are also studies showing that it may be. So maybe it depends on the parameters, but serine maybe, but the other amino acids are usually more.
So we know there is this block. And just before I move on to the next question, to answer your very first one for threonine. Threonine, we dismissed at the beginning, but also with our second metabolomic study, we could not find, it did not replicate. And neither it did replicate with other more targeted study that we have done, in which we did include threonine. It wasn't replicated. So maybe it just came up in that first study and the genetic did not confirm it, and then it kept not replicating. So we were like, we just let it go.
Like, if you provide threonine next to
would it help?
It may generate glycine.
And
it may solve the problem.
And help. It's a good idea. Unfortunately, I don't know enough about metabolism and cell metabolism to address this question more like would it help? I believe you. It's a good suggestion. Yeah, we may look into it. Yeah. Thank you. Joe.
Yeah. You didn't mention very much about the cell type in the eye that you think might be affected, other than cell death.
I did not enter.
QTL experiments or transcriptome experiments. cells this gene might be working?
So we know from histological studies that Muller glia is probably one of the first cell type in the retina to be exposed and to suffer from this. We have noticed like a reduction and a glial defect, especially Muller glia defect in the retina. EQTL and cell type specificity studies were not able yet to pinpoint or to confirm this. Well, I mean, this is already being confirmed, but unfortunately, the genomic study were empowered enough to actually confirm it from a genetic point of view. So we could not confirm that. However, a very interesting study that we have done, which I've not presented today, was a study in which the mice that were placed into a diet of serine and glycine depletion, we did actually look at their retina in terms of expression. So we'll look at single cell expression and bulk expression into their eyes. And what we notice is actually that the first tissue that was exposed is actually the RPE and not the retina. So we believe, and probably because it's so close to that retina blood barrier, is the first one to suffer from this systemic defect that is happening, which then may be affecting later on other cell types which rely on it in the retina. So we still don't know exactly what's going on. All of the experiments though that we do even in organoids keep pointing to Muller glia. So it's we are quite confident on the Muller glia component that start happening in the retina, which then may degenerate then to a loss of photoreceptors, which however happens much later on in the disease. I have one question from Zoom. Since metabolites don't have cis, trans the same way genes or protein do, how do you account for horizontal pleiotropy in your Mendelian randomization study? For example, could serine and glycine have common genetic drivers? That is a fantastic question. Yes, we did account for horizontal pleiotropy in different ways. So we look, of course, at the classic tests, for example, like MR-Eager test or other tests for the assumption that we make on the instrumental variables. Serine and glycine could have common genetic variants, absolutely. And dividing the two is really hard because, again, one can be made from the other. So truly dividing which variants are only in serine and are only in glycine is a little bit tricky. We can use what we know from the metabolic pathways and which enzyme are involved in each of them. But yes, as you say, it's very, very tricky. One way that we tried to do was we performed this Mendelian randomization and we conditioned for both glycine and serine at the same time. So we tried to test whether they were both significant if we included both in the same model. And the answer was yes, they both remained significant. However, all of the SNPs that were affecting serine were highly affecting MacTel, but not all of the SNPs that were affecting glycine were affecting MacTel. And serine fits much better than glycine with this story. However, we think that one causative mechanism in MacTel is actually an extreme depletion of glycine because of the urea cycle. And serine is trying to compensate for this lacking glycine. Hence, glycine is low, serine will be pulled down, but again, it's the pulling down of serine which then causes these cascades of effect, resulting in the oxysphingolipids. Yeah. Any other question? All right.
I have one more, but I don't want to close.
Yeah, sure. No, of course.
So
yeah.
my question is, as you mentioned, that there's significant overlap between the D and MacTel. So the question is, I assume that people tried the diabetes drugs, and more narrowly those addressing the vascular issue and degenerative issues, diabetes, to treat MacTel.
You know that I don't know. I don't know the answer to this question. I know that back in the day, one treatment that was used, which is used in AMD to treat the vascular defect that happens later on in MacTel cases. MacTel cases present with neovascularization towards the end stages of this disease. And they tried injection of VEGF which is like anti-VEGF. That not only did not work, but as much as I remember, it made things worse. So we don't know exactly. Now, about metformin, I'm not sure. Would, for example, like treatment of metformin in MacTel patients with type 2 diabetes help on the progression of MacTel? We have not performed such a study, or not that I know. Maybe someone did, but I'm not aware.
Okay.
Cool.
That's the speaker one more time.
Thank you so much, everyone. Thank you.